your prodrug, like the name's in gray, it's something that looks roughly like that. A sugar, up, at position 1', there's the base. For example, C for lamivudine or emtricitabine. And then at position 2' of the sugar, there is no OH, so it means it's a deoxyribose for DNA. And then importantly, at position 3', there is no OH either. Now, we'll see why that's important. We don't care about position 4', but on the 5' position, there, what's important is this is where we're going to attach the phosphates. And we're going to put 3 phosphates. Kinases add the phosphates to make a nucleotide. You recognize nucleotides now? So a base attached to the 1' of the sugar, and then there's no 3' OH. Why is that important? Because normally if I put the next nucleotide right here with a base, well, normally the bond is made between the 3' OH and the 5' phosphate of the next nucleotide. So I'm going to say here, can't make the phosphodiester bond. I cannot make the phosphodiester bond with the next nucleotide. Hence, chain terminator. You okay with that 3' OH something, Zaire? Is it clear? Is it clear for everyone? It's a big group of drugs, those chain terminators. Really, some of the major antivirals we have, that's exactly the way they work. And they just simply don't have a 3' OH. So you know, a very famous one for this is dideoxyinosine, DDI. And dideoxy is dideoxy because there's no OH at 2'. It's a deoxyribose. There's no OH at 3'. So it's also a chain terminator. Now, what they can be is another group, like maybe an azido group for azidothiamidine. So why is the phosphorylation necessary for it to be a nucleotide? Doesn't it already have no 3' OH? So Zaire, it doesn't have a 3' OH, so that once it's inserted into the DNA, you won't be able to attach the next nucleotide to it. But before you can insert into the DNA, well, DNA polymerases, they need nucleotides as substrate, not nucleosides. So first you need to have phosphorylation of the nucleoside drug to a nucleotide with 3 phosphate, so that then the polymerase removes 2 of these phosphates to make the phosphodiester bond. Is that okay? 2 of these phosphates will have high energy. So that's to insert the drug in it. And it does mean that at the end of the DNA of the virus will be the actual drug. So that's what we call it, a chain terminator. You can't put the next nucleotide. So meanwhile, in here, I gave you entecavir. Entecavir is a guanosine analog. So not a C analog, but a guanosine analog that inhibit DNA polymerase, but it's only used for HBV. For lamivudine and emtricitabine, these are truly nucleotide analog that block DNA polymerases. Tenofovir, that's the only one that's already a nucleotide. It's still a prodrug because it's a complex salt, but it's already a nucleotide. It already has the phosphate attached to it. Otherwise, it's still a chain terminator. For nevirapine, that's what we call a non-nucleoside reverse transcriptase inhibitor. So it's only for HIV, yeah? And I'll tell you a little bit more about hepatitis C. So for right now, what I do want you to tell me before we take a little break and then go more into liver stories, obviously, quite importantly, why can I use drugs for HIV also to treat HBV when they are reverse transcriptase inhibitors? So let's put a note right here. NRTIs for HIV can be used in HBV. Why? Because, tell me. HBV also has a reverse transcriptase. Absolutely. HIV, it's a retrovirus. It's positive single-stranded RNA diploid, and it uses reverse transcriptase to make a cDNA, and then uses host DNA polymerase to make a double-stranded cDNA, and then it uses integrase to put in the human genome and stably integrate the double-stranded cDNA inside the human genome. Now, for hepatitis B, that's not a retrovirus. Hepatitis B, it's a partially double-stranded DNA virus. It's circular DNA. And interestingly, it first uses an RNA polymerase to make an RNA intermediate, and then it uses reverse transcriptase to make cDNA. And then, of course, have some gene expression of key proteins like surface antigen, core antigen, E antigen, and so forth. So because of this, then you realize, well, both have reverse transcriptase, even though they're very different viruses. So the least toxic NRTIs are also used in HPV. And these are the three answers I gave you here. So answers B, D, and H. So I'll put right here B, D, and H above. So know them well, because it does mean that if you are using these drugs for HIV, make sure you double check whether the patient also has hepatitis B. Because if you were to remove the HIV drug, maybe for a side effect, a drug interaction, or something of that nature, then unfortunately you would have a flare-up of hepatitis B in the patient, and you would trigger a fulminant hepatitis. So always when you use NRTIs, ensure whether or not there's a positive serology for hepatitis B. Well, let's see. Zaire, you have a question? Can an RTI act on other DNA-RNA polymerase? And if no, why? So, Zaire, NRTIs can work on one important DNA polymerase in the human cell. The most primitive DNA polymerase we have is the one we have in our mitochondria. It's called gamma DNA polymerase, and unfortunately, it is fooled by the structure of the nucleoside reverse transcriptase inhibitor. The other DNA polymerases we have, they don't pick up the drug. They are so specific for the structure of a nucleotide, they're not fooled by the drug. Is that okay? So your alpha, your beta, your epsilon DNA polymerase, they're absolutely untouched. Only the gamma DNA polymerase of mitochondria. These drugs cannot block RNA polymerases. The reason why? They are based on deoxyribose, not on ribose. So obviously RNA polymerases, they would recognize riboses. They would have an OH at position 2'. Does this answer both your important question there? Right. So you would need a ribonucleotide analog to block RNA polymerases of RNA viruses. And other DNA polymerases, particularly of eukaryotic cells, they are far too specific for the nucleotide structure to be fooled by the fake structure of a drug. Now, if there's mutation to the structure of the reverse transcriptases, they may not work. But here's the deal, Zaya. That's why we have NRTIs of all 4 different bases. We have NRTIs of T's, like zidovudine, stavudine. We have those of C's, of cytosine, like, of course, emtricitabine, lamivudine. We have those, of course, of A, like, for instance, didanosine, like tenofovir itself. And then we even have an analog of C, which is abacavir. And when there's a change in the structure of reverse transcriptase that causes resistance to one of the drug, generally it doesn't to the other. So we have good chances. Yep. But now the ones we use for hepatitis B have to be the ones that are least toxic. And those are the 3 I gave you. The others have too much of these mitochondrial toxicity to be considered good, for that matter. So while we have this, we have important drugs for hepatitis B. You have also the guanosine analog entecavir, and then you have the famous interferon, which are simply doing what we're supposed to do, which is enhance our immune response. Let me put a